MSc MPhil PhD to deliver the welcome address.
Thank you, Radha. So, good afternoon, one and all present here. It gives immense pleasure to propose welcome address to this guest lecture. So, under DPT Star College scheme, today we have guest lecture on Bioprosepcting of Ascidians, delivered by eminent research personality Dr. H. Abdul Jaffar Ali, Head and Assistant Professor, Department of Biotechnology, Islamiah College, Vaniyambadi. First of all, I have decided to give few lines about his career excellence, but his profile is loaded with academic milestones. So in different fields like research, projects, guideships, paper publications, paper presentations. So it is a long list. I cannot read out fully because it will take much time to give his research excellence in Ascidians. So I want to say more about his research experience. His research area are taxonomy and diversity of Ascidians, DNA barcoding of Ascidians, natural products of Ascidians, bioremediation of heavy metals in marine ecosystem. He has a teaching experience of more than 20 years of both UG and PG. His research experience is above 15 years. He is a consultant of DNA barcoding of marine animals, bioassay, antimicrobial, antioxidant, and the antimitotic assays. So he deposited 310 gene sequences covering 61 species of Ascidians in GenBank, NCBI, and BOLD. He received many awards. He received Scientist of the Year 2019 by Culture, Educational and Welfare Society, Uttar Pradesh at Bangkok in Thailand. He got Oral Presentation Award in the international conference held at Bangkok, Thailand. And he is an outstanding scientist in marine and science by Ijrula Academy, Trichy. Har Govinda Khurana Memorial Award for Zoology in 2016 he got from Bose Scientific Society, Tamilnadu. He is a special invitee in dissemination workshop at New Delhi and expert committee meeting on invasive alien species at National Biodiversity Authority, Chennai. And he got junior research fellowship from the Ministry of Environment and Forest in the Government of India in the year 2000-2002 in the project titled Marine Biodiversity and Taxonomy of Indian Ascidians. He got guideship in biotechnology from Trivulvar University, Vellore, and Bharatiyar University, Coimbatore, and got guideship in zoology from MS University, Tirunelveli. He produced 5 PhDs and two members are pursuing PhD under his guidance, and he finished 14 MPhil theses and 12 MSc projects, 10 BSc projects. He published more than 60 papers in national and international journals. He published books on microbiology lab manual, apiculture, Ascidians in hostel water, DNA barcoding methods and protocols, then Uyirin, Palamoligalil, Arvial Unmai. And he contributed chapters in books also at international level. He organized webinar, seminars and workshops. He organized 4 workshops and 2 seminars. He presented many national, international and state level seminars, presented papers in many national and international seminars. He acted as a resource person in national level webinars, online certificate course, student exchange program, and judge for interschool science fair, chairperson in international conference. He published more than 60 papers, and he is a member in UG and PG Biotechnology Board of Studies, Islamiah College, Vaniyambadi, and UG, PG, MPhil Board of Studies in St. Mary's College, Thuthukudi, and he is a member in Research Committee in Islamiah College, Vaniyambadi. He is a member in Committee for Special Schemes under DPT and member in Editorial Board in International Journal of Science and Humanities. And he is MPhil thesis examiner in Kerala University and MS University, Tirunelveli, and the question paper setter in Islamiah College, Vaniyambadi, DKM College, Vellore, KS Rangasamy College, Tiruchengood, St. Joseph's College, Kadalur, Sarada College, Salem, and St. Mary's College, Thuthukudi. So, we today have a very young, talented, enthusiastic, research-minded personality in our midst. So, on behalf of our managing board, principal, and staff members and students of our zoology department. I welcome you, sir. Really, we are fortunate enough to have such a person in our midst. So, once again, I welcome you, sir. And also, it is a pleasure for me to welcome other members from Vaniyambadi and our staff members and students to this guest lecture. Last but not least, we thank members of DPT, Government of India, to give this opportunity to enlighten our students' mind by conducting this type of guest lecture by providing us financial assistance. So once again, I welcome you all, sir. Thank you, sir. Welcome, sir.
Thank you, ma'am. Thank you, ma'am.
Thank you, sir. Thank you, sir. Okay, sir. You can proceed, sir.
Yeah, may I start now?
Okay, sir. Okay, sir. With pleasure.
Is my voice audible, ma'am?
Yes, sir. Yes, sir.
Yes, sir.
Audible, sir.
Yeah, okay.
Dr. H. Abdul Jaffar Ali, welcome you.
Okay.
Yeah, thank you, Dr. Tamilnadu.
இதுவும் நல்லா இருக்கு. Okay,
Okay,
sir.
okay.
Okay, sir. Okay.
In the name of Allah, the most beneficent, most merciful, most respected Dr. Jayarani, Head of the Department of Zoology, Vanniaperumal College, Virudhunagar, and most respected and my dear close friend, Dr. M. Tamilchelvi, the coordinator and member secretary, and most respected Dr. Radhalakshmi, co-coordinator of the Department of Zoology for the DPT Star College scheme, and participants from the various department of Vanniaperumal College for Women. A very good afternoon, one and all, to all of you present here. At the outset, I would like to thank Dr. Rani, the head of the Department of Zoology, for having given me the opportunity.
This call is now being recorded.
To present a topic on the title Bioprospecting of Ascidians. Thank you, ma'am, for having invited me. And my special thanks to my friend Dr. Tamilchelvi, who is always with me in the field of research, publications, book publications, and as a family friend too. Thank you, Radhalakshmi. Thank you, Tamilchelvi, ma'am. And I thank the Radhalakshmi ma'am also, who was my student. She did PhD under my guidance. I'm very proud to say this. Thank you, Radhalakshmi ma'am. And I thank
you,
the entire the team who is working for the DPT Star College scheme. Okay, now let me go into the actual presentation in the title Bioprospecting of Ascidians. Now I would like to share my presentation now.
Yes, sir. Yes, sir. You can share the slide.
Okay. Krishna. Okay. Sharada? Yeah, hello, ma'am. My
Yes.
slide visible?
Visible,
Yes,
sir.
sir.
Visible, sir.
Yes, sir. Yes, sir.
Right.
Visible, sir. Visible,
Yeah,
sir.
right. Okay. Kind of.
Yes, sir.
Yeah, okay. The title is The Bioprospecting of Ascidians. Before going to the bioprospecting aspect of Ascidians, I would like to give something basic about the Ascidians. How many of you have heard the animal name Ascidians? I hope you all belong to the zoology department, so you might have heard the term Ascidian And in your first semester or in second semester, you might have studied under the title invertebrata or under the vertebrata group. So the Ascidians, suppose if you have not heard about the animal Ascidians, some of you
In the second semester, sir.
should have heard the term Ascidian because the faculty, Dr. Tamilchelvi, is doing enormous work on the Ascidian, and under his guidance, one more faculty from your department is also doing PhD in the field of marine Ascidians. So I hope definitely some of you from your zoology department might have heard something about the Ascidians. Okay. So why we should go for the bioprospecting of Ascidians? That is the core and the prime concept of this presentation. Right. Before going to detail about the Ascidians, let me brief about the bioprospecting. What does it mean? Bioprospecting may be otherwise called a biodiversity prospecting.
This call is now being recorded.
Yeah. So what is bioprospecting? Bioprospecting is nothing but a targeted exploration and a systematic search of plants, animals, and microbes for new or better products, genes, or technological processes. So this is what we call bioprospecting, systematic searching for new bioproducts or better products or some new genes or some technological processes from plants, animals, and microbial species. So this is a bioprospecting. Then why we should go for the bioprospecting? So from the definition itself, you might have understand the purpose of bioprospecting. The first thing is to find new resources, new bioresources, and new bioproducts from the nature, and that can be used for the welfare of the human being, and to improve the human health by exploring the novel drugs and also protein-rich nutrients from the living organisms in the nature, and also to discover the lead molecules for the development of various drugs. So these are the prime reasons why we go for the bioprospecting, because nowadays we are using so many products right from the morning wake up to till go to the bed. Say for example, the morning we use the toothpaste. So toothpaste, it is a product obtained from the some kind of organisms or animals or plants. Then we are using so many facial creams prepared and manufactured from using the biological organisms. Until when you go to the bed, we switch on the that is mosquito liquidator. So that also produced from the various organisms. So in the day-to-day life, we are using the various products which we obtained from the biological origin. Apart from these day-to-day life products, some other very important novel products which we get from the natural resources. Say for example, see, so this is a picture, that is a fluorescent microscopic view of a bacterium called Thermus aquaticus. You may be aware of this bacteria, I think so. Thermus aquaticus. It is a bacteria which can withstand high temperature. So the enzymes, the proteins which are synthesized by this bacteria are also resistant to the high temperature. So this one important enzyme from this bacteria called DNA polymerase. You may be knowing this. So the DNA polymerase is a very important findings in the biotechnological aspect, and this DNA polymerase is used in the important process, important technique called what is that? The PCR, that is polymerase chain reaction. So polymerase chain reaction is a very important techniques, reactions discovered in the presence of the DNA polymerase. With this DNA, that is the polymerase chain reaction, so many biotechnological processes, so many recombinant technologies, and many biotechnological processes products are developed. So you may be knowing that different diverse applications of polymerase chain reaction. So this is the one product, enzyme, bioenzyme, that is DNA polymerase from Thermus aquaticus. Similarly, another one. So this is a fish which is present in the Antarctic ice region. So the protein synthesized and present in this fish is called antifreeze protein. And this antifreeze proteins are used in various technologies and various processes and even the development of the drugs. And one more important findings from the bioprospecting of marine organism is a green fluorescent protein. This protein was isolated from the jellyfish, Echoria victoria. And this fluorescent protein is nowadays used in the goldfish for the development of different colorings. And see, this is the one paint, load to sun. It is a paint which is resistant to water, resistant to the bacteria and fungus. This paint is manufactured by the natural phenomena which occur in the leaf of the lotus plant. You may be knowing that, the water molecules will not attach to the leaf of lotus. So there is a process and the arrangement of that lotus leaf which made the water into a droplets or a spherical sphere, and only few surface area of the water is allowed to attach with the leaf. And this technology is mimicked and used in the preparation of paint, and now this paint is used in various purposes like painting the utensils, painting the vehicles, painting the buildings. So this is a bioproduct. And this is another one product developed from the biological organisms. It is nothing but a mat. And see, this is chapel. These two products are fire resistant products and developed from using the
Okay, you can continue, sir.
right. Okay.
Okay, sir.
Yeah, right. Okay. ஸ்டார்ட் பண்ணியாச்சு. நல்லா தான் பண்ணிட்டு இருக்கிறோம். இல்லை தெரியுது இல்லையா? இது வருதா? இதில் போடணுமா?
Presentation வரல, sir. PPT வரல, sir.
Wait a minute, ma'am.
Okay, sir. Okay, sir.
No problem. Yeah, okay. Now visible?
Okay. Yes, sir.
provide new surgical adhesives also. And see, this is the oil pollution in the marine ecosystem. So you may be very much aware of the bacteria. It's a oil eating bacteria. So the invention of this bacteria is utilized to digest all the oils in the marine ecosystem. See, this is the bacteria name is Alcavinorax borkumensis. It is a very small bacteria that thrives on the petroleum or in oil. Why?
Sir, PPT. Okay, sir. Okay, sir.
Right.
Now it is okay, sir.
So this bacteria thrives on the petroleum and oil environment, and this can be feed on the both hydrocarbons and alkanes which are abundant in oil. And this bacterium occurs naturally near oil seeps in marine ecosystem. And this is one more important product, that is limbless amoebocyte lysate. This is obtained from the Atlantic horseshoe crab, and the gel is prepared, and this gel is used in the microbial assessment test, that is called a gel clot assay or gel clot test. And this test is used to detect gram-negative bacteria toxins. It's a very important breakthrough in the field of medical. So see, this is the horseshoe-shaped crab. So from the blood of this crab, a liquid is extracted, and this liquid consisting of the limbless amoebocyte lysate, and this is used to detect the gram-negative bacterial toxins. And various enzymes derived from the extremophiles have been used in detergents, food processing, cleaning, dyeing, medical diagnosis, skin protection products, and forensics. So many enzymes which are currently available in market for various purposes. All these enzymes are nothing but the bioproducts due to the bioprospecting technology. Okay, then where to bioprospect? In which area in the globe? Where to search? So that is very important. Where to search? See, so wherever there is a rich biodiversity, we can go for the bioprospecting, so that we will get the new species of plants, animals, or microbes. Through this, we will get the new products or genes. Particularly, the biodiversity in extreme environment and the unique ecological niches, we have to select the extreme environment and a very unique ecological niche for the exploration of the various organisms. And through these organisms, we will be able to search for the or produce the various bioproducts. But only tiny fractions of the world's biodiversity has actually been explored. So you may be knowing that there are, it has been estimated that more than 10 million to 100 million living organisms are existing in the earth. It has been estimated approximately, out of which only 2% of the natural resources in the terms of living organisms are utilized, and rest are not yet explored and not yet utilized. And the richest area of biodiversity, which tend to be in developing countries. So we have to select the untapped resources, in particular the extreme environment and the unique niches, so that we will get the new species of plants, animals, or microbes, and we will get the new bioproducts or biotechniques. and the extreme environments which provide habitats for the extremophiles. So the organisms living in very extreme conditions or extreme environments with unique characteristics which developed for the survival. So extreme conditions like in the very deep sea or in the high altitudes or in the icebergs or in the high saline soil. So these are some extreme habitats where some unique organisms will be there with unique characteristics. The biological processes and the materials which enable these extremophiles to survive in the extreme conditions. So those animals which can survive in these extreme environmental conditions can develop the different biological processes and the products to withstand these extreme environments, such as temperature, pressure, salinity, etc. And other unique conditions are the sources of great potential for scientific advancement and commercial applications. Among the extreme environment, the ocean environment is a massive complex one consisting of extreme variations in physical parameters and biological parameters such as the pressure, salinity, temperature, salt content, and ocean currents, and biological habitats like deep sea, seabed, coral reefs, etc. So the bioprospecting of marine environments is conducted almost exclusively in regions at extreme depths, specifically around submarine trenches, cold seeps, sea mounts, and hydrothermal vents. Actually, the natural products isolated from the marine sources tend to be more highly bioactive than terrestrial counterparts. So there are so many bioproducts which are obtained from the terrestrial organisms. But when compared to the bioproducts and the biotechniques which we obtained from the marine sources are highly bioactive than the terrestrial ones. All because of the extreme environmental habitats present in the ocean environment. And now we will go to the bioprospecting approaches. How we should approach the bioprospecting of various organisms. So one is the random approach. Here the collections can be made on random within a geographical area to obtain the representative samples of the local biodiversity. So random collection within a geographical location so that we can obtain a representative samples from the particular local biodiversity ecosystem. And the second approach is chemotaxonomic approach. Here the collections of organisms are based on the knowledge of taxa with certain classes of compounds of value to research and screening. So we will be knowing that so these organisms may contain this kind of products, like for example herbal plants. So we are aware that there are some herbal plants and the compounds which are present in that herbal plants. We know the applications, we know the compound. So we have to target that one for the new products or better products. And another approach is ecology driven or biorational approach. Here the collections are based on the ecological relationship between the species. Different ecological relationship in the case of different ecosystems or in the ecological succession regions where you can collect the diverse form of organisms. And another one is the ethnobiological collection based on the indigenous communities. So the native species, collection on native species of plants, animals, or microbes. So these are the 4 different approaches through which we can bioprospect the new organisms or new microbes. Then the process of bioprospecting. So what are the steps involved in the bioprospecting? There are 4 phases. The one is on-site collection of samples. So we are going to the selected spot or the extreme environment or whatever may be the environment. There is a site, collection site. We have to choose the collection site. After that, isolation, characterization, and culture of the specific compound. So once we visited the site, then we will collect the samples. So after the collection of the samples, we have to isolate the compound which are present in these samples collected. Then we have to characterize the compounds and we have to develop the specific compound. And the phase 3, here we have to screen the products which we isolated and screening for the potential uses. Say for example, pharmaceutical uses or nutraceutical uses or any other uses. And the fourth phase is the products development and the commercialization. Once we developed the product and screened for the potential uses, and if it is okay, then it should be commercialized. So fourth phase is the commercialization. And once commercialized, the products can be applied for the patenting and sales, then marketing. So these are the 4 different phases of bioprospecting. And the major areas of bioprospecting: the chemical bioprospecting. Here you can develop the drugs, pharmaceuticals, pesticides, cosmetics, food additives, and other industrial products. Then gene bioprospecting, genetic engineering, development of the various recombinant genes and clones. Then development of hybrid crops, then fermentation techniques, then cell cultures. And another area of bioprospecting is bionic prospecting. So here we can develop the designs or the technologies or the processes, the architect nature, bioengineering, biomodeling, all these processes which we can develop from the living organisms. I told you the development, the construction of the new building, a very tall building which mimicked the termite mounds. So this kind of process that can also be developed using by bioprospecting technology. So this is the graph which clearly shows that the number of articles describing the marine natural products has increased in the last 50 years. You see, number of marine natural products and number of bioproducts from both plant and marine resources. So the number of articles has been increasing, but it remains significantly lower than those describing the plant natural products. Even though large number of marine natural products are available in the marine ecosystem, the products or the research or the articles related to the marine natural product is comparatively less. So we have to go for bioprospecting marine ecosystem, right? So apart from this potential applications of bioprospecting, on the other side of the coin, that is some environmental impact also we are facing during the bioprospecting. This is a negative aspect of the bioprospecting. What are the environmental impacts? See, this is the in situ experiments in deep seabed. Already I told now we have to search the deep sea, or we have to go search in the extreme environmental conditions. So when we go for searching new plants or new organisms, we are using so many technologies. So here in the deep sea, the scuba divers using various equipments for the collection and sampling of the organisms. So they introduce the high intense light and also the noise. So these high intense light and noise will change the water temperature that will affect the survival of the organism. And see here, this is the waste or the debris which are discharged from the vessels or the equipments which we use for the collection of samples in the marine environment. So the discharge of waste and debris that cause the marine pollution. And see, this is the discarding of scientific samples. Sometimes we sample the fishes or any other organisms from the marine ecosystem indiscriminately. So unwanted or additional or excess bio samples we are discharging into the same habitats. So that also causes the biological contamination. And here this is the over exploitation. So we collect these samples indiscriminately, and excess amount of or excess number of organisms we are collecting for our purpose. This is over exploiting. So due to the over exploiting, particular species may go extinct. So these are some of the impacts of bioprospecting. Okay, now come to the marine natural products. So MNP is nothing but marine natural products. Nearly more than 20,000 marine natural products are isolated from various marine organisms, particularly from the sponges, from bryozoans, from tunicates, from soft corals, from nudibranchs, sea hares, sea slugs, and from other marine organisms. So these organisms are the major producers of marine natural products with a significant biopotential and biopotential activities. And in this scenario, the marine ascidians or the tunicates are gaining paramount importance in remaining a vast untapped resource for the bioactive compounds. And the ascidians represent the most highly evolved group of marine organisms which are commonly investigated for identification of marine natural products. And these ascidians provide rich source of bioactive secondary metabolites with promising potential applications, biomedical applications. So now we will go into the bioprospecting of ascidians. Right. So let me give a brief introduction about the ascidians. What are ascidians? So ascidians, otherwise called tunicates or sea squids, they belong to the class Ascidiaceae of subphylum Urochordata, or otherwise called tunicata. They form an important group of marine benthic sessile communities, and the body is enclosed in a test or a tunic, which is a body covering, similar to the shells in the molluscans. In the molluscans, you can see the hard cells inside the shell. The animal will be there. Similarly, in ascidians, a tough, leathery covering will be there. Inside the covering, the soft-bodied animal will be there, and this covering is called a test, or otherwise called a tunic. Hence, the name tunicata. All ascidians are a sessile one. Sessile means they are sedentary. They cannot swim, they cannot move. So they will be in the same place till their death. But the larval form is planktonic. Larval form is motile. They can move, they can swim. Nearly 3,000 described species found in almost all marine habitats from shallow water to deep sea. They are cosmopolitan in distribution. They are exclusively marine. No freshwater forms. And ascidians, otherwise called tunicates, I already told you, because their bodies are enclosed within a tunic, which are made up of cellulose-like material, tunicine. So this tunicine is a very rare occurrence within the animal kingdom. Only in the ascidian, this tunicine is available, present, and this tunicine is nothing but a cellulose-like materials. Cellulose will be available. Will be present only in the plants, not in the animal. But in the ascidian, the cellulose-like material called tunicine is present. So this is the tough, leathery covering of the ascidian. See the picture. And it is otherwise called a sea squid, because these ascidians, they pump the water through their branchials or the atrial siphon once the animals are removed or disturbed. So look at this picture. So one man holding the ascidian. So when we disturb the ascidian or when we touch the ascidian, some amount of water will be pumped out. That is, it will squid the water. That is why it is called a sea squid. See, so when we touch or when we disturb the ascidian, it will pump out some amount of water. Right. So this is the main identification, field identification characteristics of Ascidians. Right. Types of Ascidians. There are three types of Ascidians. The first one is a solitary Ascidian. It is a single, a discrete individual. You look at the picture, you can see a single individual with two siphon. One is branchial siphon. It may be referred as a mouth, and this is the atrial siphon. It may be referred as a anus. Okay, and this is the life cycle of solitary or single Ascidian. This is the larva. It is in a tadpole larva. It is a motile. Once the larva find the hard substrate or suitable substrate for their attachment or settlement, with the help of some adhesive papilla, this larva will bind with the substrate and they start developing. They will undergo the retrogressive metamorphosis. It is a characteristic feature of Ascidian, and it start developing and it becomes adult. So once it attached with the, any substrate, then they will not move, they will not swim, they will be as such till their life. And the second group is the compound Ascidians or else colonial Ascidians. Here the individual zooids are very microscopic, and so many zooids are integrated into a gelatinous matrix to the extent of sharing a common excrement siphon with the neighboring zooids. You see, this is the ball-like structure. It's a gel matrix. Inside you can see the small dots like structure. It's nothing but a individual zooid. So you see, and with a common opening through which the respiration and excretion will be taking place. And see, this is the one zooid structure. It is a microscopic view of a single zooid of colonial Ascidian. Okay, this is a microscopic view. So this is the larva of colonial Ascidian, larval structure. And this is the life cycle of the colonial Ascidian. You see, this is the colony of the Ascidian, where you can see the number of zooids will be there, and they produce the larva, motile larva. Then this larva finds the hard substrates and start attachment. Then it will settle on the substrate. Once settled, they start growing, they multiply, they grow in larger size. Then again they will come to adult. Then the life cycle will go on. And another type of Ascidian is called a social Ascidian. It is nothing but simple Ascidians, but few members of the same species, they join together in a through a stolonic vessel. So here, this is the one single Ascidian, this is the one single, this is another single. They belong to same species, but they are attached through some stolonic vessels. So this is called social Ascidians or synascidians. And the Ascidians, they are morphologically diversified from microscopic to macroscopic. See, this is a microscopic, very small Ascidian. They are grouped, and here this is the single individual. It is a very large one. And here this is also a red colored one attached on the surface of the stone. This is a thin sheet. It will be in thin sheet form, and it will be in a globular mass form. It will be in a lengthy form. It will be in a very small size. And here you can see there are so many Ascidians which are colorless, whereas some Ascidians are deeply colored. You see, this is the colorless colonial Ascidian, this is the deeply colored colonial Ascidian, and this is the colorless solitary Ascidian, and this is a deeply colored solitary Ascidian. And these are some of the vernacular names of various Ascidians: dark Ascidian, red throat Ascidians, orange throat Ascidians, like that. So there are so many vernacular names for the different species of Ascidians. These are the examples for the solitary Ascidians. You can see the two siphon. This is a two siphon, one siphon, branchial siphon, this is a atrial siphon, this is the whole body. This is a transparent Ascidian, it's a dark black Ascidian, so colored Ascidian. So you can see the samples of the simple Ascidians. And this is the examples for the colonial Ascidians. You see in the left hand side, it is a very glassy colonial Ascidian. In the right hand side, you can see colonial Ascidians, particularly the diadem species. And this is also an example for the colonial Ascidians, a moss-like structure. And you can see the white color, a sheet-like structure, which is on the surface of the embedded rocks or stones. See the white colored one. And this is the glassy colonial Ascidian. Okay. Now the settlement, Ascidians, where they settle, that's very important. Most of the Ascidians, they settle on the hard rocks, then stones, the hulls of ships and boats, the branches and roots of tree and algae which are present in the coastal area, and they settle on the various floating objects like ropes, rafts, barrels, and nets which are used in the harbors, and other harbor installations like pilings, large barges, poles, cement blocks. So these are all the substrates. On this substrate, the Ascidians settle. There are two types of substrates, soft substrate and hard substrates. So in all kinds of substrates, different forms of Ascidians, they settle. Say this is a large embedded rock, which is in the near Valli cave, Tiruchendur, where you can see at the bottom of the rock, we collected so many samples of Ascidians. And this is the iron pipes which are used in the Sekri Tuticorin region, and on the iron pipes which are immersed into the seawater for some other purposes. So on the surface of the iron pipes and materials, we can see the red colored one. They are nothing but the colonial Ascidians. Similarly, in the small stones, the whole entire stone is covered with a thin sheet of Ascidians, red color here also. And it is a pillar of the jetty where you can see the settlement of Ascidians. And this is the iron pipes and some materials which are used in the boat or in the harbors. So on the surface of that hard iron pipes, some Ascidians will be there. In the rope, a group of Ascidians are there. Here you can see the sponges. These are spiny sponges. Over the sponges, you can see the grape Ascidians, a green colored one Ascidians. They are called grape Ascidians. So this is called encrustating Ascidians. So the Ascidians attach on the surface of the other animals like sponges or molluscans. You see the encrusting Ascidians over the sponges. So here also the sponges, over the sponges you can see the group of colonial Ascidians. Here the embedded rocks where you can see the different forms of Ascidians, red colored, black colored, like that. And on the shell of the bivalves, you can see this is the bivalve shell. So in the dead bivalve cells, you can see the attachment of Ascidians. See, this is the dead bivalve cell which we collected from the bottom of the sea, where you can see the formation of white colored colonial Ascidian. So here the encrusting Ascidian over the sponges. So here the embedded rock in the shallow water region, where you can see this is the dirty colored one. It is nothing but the colonial Ascidian, Diadenum species. Here also you can see the white colored lobed colonies of Ascidians. So the nets which are used in the different cages, in the cage culture, you might have seen the cage culture of crabs, cage culture of lobsters or some other marine molluscans. So in this ropes of the cages itself, you can see the settlement of some Ascidians. Here in the chanks, dead chanks, you can see the attachment of Ascidians. So this is the pillar of the jetty used in the harbor, where you can see the settlement of Ascidians. This is the hull of boat where you can see the biofouling of Ascidians. You see, this is a small rock. This is nothing but Dr. Tamilnadu is collecting the samples from the shallow water region, and this is a dead shell, molluscan shell. Inside the dead shell, you can see the presence of Ascidian. Then distribution. Ascidians are distributed throughout the world oceans, from the tropical water, temperate regions, and even within the cold and subpolar waters. And this is the data which represents the number and percentage of the species per order. So there are three orders: Blephobranchia, Stolidobranchia, and Aplosobranchia. This Aplosobranchia represents the 50% of the Ascidian species. So this is just data regarding the Ascidian species. And this is a graph which shows the number of species by family within each order. You see the family, so here the different families and the number of species represented. So Aplosoprancha 1480 species, in the Blyphobrancha 315 species, and in the Stolidobrancha 1020 species so far recorded throughout the world. And this is the graph which shows the percentage and number of species described per taxonomic authority. A renowned world taxonomist, they have contributed much in the exploration of the various Ascidians. So Monit and Monit 20%, Coat 16%, and all others. And from the India, and myself and your faculty, Dr. Tamilnadu, we are contributing 2% of the Ascidian representation to the world database. So this Ascidian global distribution is a map which shows the species in the various countries. And the diversity of Ascidians in India has been studied over the 2000 kilometers of the coast of the India, right from Bombay to Visakhapatnam, encompassing the various regions such as Arabian Sea, Indian Ocean, Gulf of Mannar, Pak Bay, Bak Strait, and Bay of Bengal. So in all these regions, the Ascidians have been reported during the various period of time. This is a history of Ascidians. Ascidian was first reported by the Vodka during 1915, and there are so many Ascidian taxonomists. They explored Ascidians in various period of time. Totally 400 species so far reported from the Indian coast. And identification method, how we identify the Ascidians and what methods we follow to collect the samples, all are given here. Once they collected, what next we should do, identification, preservation, all these things we will do while we collecting these species and recording it. The collection method, we can hire the snorkelers to collect the Ascidians from the deep sea or deep water, right from 2 meter depth to 3 meter depth, like that. And we can collect these species by scraping off method from the shells. And this is the hand picking methods present in the net. And this is a scuba diving method which we adopt to collect the Ascidians from the deep sea and coral regions. And these are some methods we adopted for the collection of Ascidians from various coastal regions. So this is the hull of boat where you can see full of colonial Ascidians occupying the entire bottom of the boat. So which we collected from the Kolachal coastal region. And my scholars are collecting these samples from various substrates. And even we collect the samples from the pillars of the Palm Bridge by adopting the snorkelers and other scuba divers. And this is the classification chart which we follow for the identification and classification of Ascidians. And these are some taxonomic characters which we observe and which we describe for the identification conformations. These are all the taxonomic characters. Now come to the bioprospecting of Ascidians for the development of novel drugs. Ascidians, the candidate species for the novel drugs. See, the one first bioactive metabolites, that is a geranyl hydroquinone. It is a marine bioactive compound which was isolated from the marine species called Aplydium. This is the graph which clearly shows that during the period 1974 to 1992, only 230 number of natural products have been isolated. Whereas from the 1994 to 2014, the number of natural products has been doubled to 572. Nearly 600 natural products have been isolated from the Ascidians. So this is the graph which clearly shows that the marine natural products studied from the various families of Ascidians. So Aplosoprancha contributed 72% of natural products, Blyphobrancha contributed 3%, and Stolidobrancha 25%. And this is the percentage representing the chemical diversity. What kind of bioactive compounds present in the Ascidian group of animals. So the very common compound is nothing but the indole alkaloids, which are very common in most of the Ascidians. So nearly 48% of the natural products, they belong to the indole alkaloids. And rest of the next one is the pyrrocridine alkaloids. Similarly, so many bioactive compounds with the chemical diversification from the Ascidians. And this is the bioactivity of the marine natural products isolated from the Ascidians. You see, 60% of the bioactive compounds isolated from the Ascidians are used for the treatment of the tumor or cancers, which is followed by the antibacterial activity. The compounds, 14% of the marine natural product compounds are used as a drugs for the as a bactericidal effect or killing the bacteria as antibiotics. Similarly, antifungal, antiviral, and even anti-HIV, anti-malarial drugs, anti-diabetic drugs, antioxidant compounds, anti-inflammatory compounds, central nervous system compounds, and all other compounds. So nearly almost most of the biological activity, that is the marine natural products which are isolated from the Ascidians, showing the different biological activities. Then why should Ascidian produce bioactive compounds? So why Ascidians producing the more number of natural marine natural products with a significant activity? So the reason, Ascidians, you may be knowing, already I told you, it is a sedentary organism and filter feeders. They are filter feeders. So during the filtration of the sea water, they accumulate high concentration of the bacteria from the sea water from endostyle to atrium. So this is the branchial siphon. Through this siphon, the water will enter into the body. Here the filter feeding mechanism will be there. So this mechanism will filter the sea water. So during the filtration, high concentration of the marine bacteria will be accumulated in the body of the Ascidian. So to get rid of this microcosm, that is to get rid of the high concentration of the bacteria, Ascidians have to produce the various compounds to fight against the bacteria. Right. So to get rid of the microbes, Ascidians synthesize various kinds of antimicrobial compounds. So we can expect the antimicrobial compounds from the Ascidians. So these are the so many antimicrobial compounds isolated from the Ascidians. And Ascidians, they are sedentary and they are good biofoulers also. You can see the biofouling Ascidians over the hull of boat, hull of ships, and even in the hard substrates present in the harbors. So this could be possible by the Ascidians because they can produce some compounds for the defense purpose, because they have to compete for the space. Biofoulers should compete for the space. So they produce the different compounds or chemical compounds which are very effective to kill rapidly dividing the cells of neighboring foulers. So the Ascidians, they produce some compounds which kill the neighboring cells or neighboring organisms, so that they can overcome the spaces occupied by the other organisms. So this nature, that is this ability of chemical to rapidly kill the dividing cells, is the hallmark of the chemotherapy. So which leads to production of various compounds with some antiviral or anti-cancer drugs. So one can expect the antiviral anti-cancer drugs from the Ascidians. Similarly, many benthic Ascidians, you see the right hand side top picture, these are the Ascidians. The test, the body covering of the Ascidians is free of any organisms. The test is very clear. No other organisms can attach with the test of these organisms. This is because these Ascidians have some mechanical defenses. Say for example, the tough tunicates, the tough membrane or the leathery membrane, and the spicules. Here the micrography photo of these spicules. There are so many minute microscopic spicules will be present inside the test, similar to the sponges. So this physical defense mechanism, that is presence of test, presence of spicules. So due to this mechanism, no other organisms can epibia on the surface of the test. Okay, so this may deter the predators. So apart from these physical or mechanical defenses, some chemical defenses also present in some kind of Ascidians. So some compounds which are secreted by the test of these organisms, and these compound reject or expel the other organisms to attach over the surface. So this property is called a anti-fouling property, and the compound which exhibit this property is called a anti-fouling compound or repellent compound. And this compound now has been utilized in so many paints. That paints are used in the painting the hull of boat and hull of ship, so that the hull of boat and hull of ship could be free of any biofouling Ascidians or biofouling organisms. Then what are the other Ascidian derived compounds? You see, the picture clearly shows, you may be aware, because nowadays we are very much aware of the COVID-19 and the coronavirus. So here, this is the picture of the coronavirus structure. The in vitro study results of litidepsin, it is a compound. Litidepsin is a compound which act on the human coronavirus COVID-229E. So this strain of COVID, that is coronavirus, has a multiplication and a propagation mechanism very similar to COVID-19. So COVID-19, it is nowadays we are facing the similar strain, that is HCoV-229E. Its mechanism, multiplication and the propagation mechanism, is very similar to COVID-19. And this COVID-19 virus was killed with the compound called litidepsin, which has been isolated from the Ascidian. It is a novel anti-cancer drug possibly active against the COVID-19. Recently, it has been reported and published. And this is another compound, Aplidin, from the Aplidium species, which isolated from the Ascidian Aplidium albicans, and first reported in 1991 patent application. It is in preclinical animal test. This compound exhibited anti-cancer properties, and also it is described as a multifactorial apoptosis inducer. This is an important novel drug from the Ascidian. And it is another important marine natural product called diadamnin B, which was isolated from the Tridabnum solidum. It is a colonial Ascidian. See, this is the white colored Ascidian. It's a very important Ascidian, very significant Ascidian. From this Ascidian, the diadamnin B is a marine natural product, has been isolated. And this is acting against the various bacterial strains. And another compound, yondidylis, it is commercially available in the market, and this compound has been isolated from the Actinosidia turbinata. It's an Ascidia. Here you see the picture. And this compound is very effective against the advanced soft tissue sarcoma, osteosarcoma, and metastatic breast cancers. And it is traditionally, commercially, it is named as ET743, yondidylis. And this has been co-developed under the trade name yondidylis by the Spanish marine pharmaceutical company PharmaMar and the Johnson & Johnson subsidiary Ortho Biotech company. And another important compound, a drug developed from the Ascidian, is the midostarin. So it is a semi-synthetic derivative of statosporin PKZ 412 midostarin, which was isolated from the bacterium, but this bacteria is also isolated from the Ascidian. There are some bacteria which will be closely associated with the Ascidians. Okay, so Ascidian associated bacteria. So the bacteria which are associated with the Ascidian is nothing but the Streptomyces saurosporus. From this bacteria, this new drug has been isolated. And another compound, anti-drug, anti-cancer compound, vitalamide, isolated from the diadenum cuculiferanum and the polysyncreton lithostratum, is one of the several novel tubulin interactive agents recently discovered from the marine resource, particularly from the Ascidians. And this compound inhibits the tubulin polymerization and can arrest the cell cycle of target cells in the G2 or M phase. So this drug is nowadays used in treating the various cancers. And this another compound, diazonamide. This also isolated from the Diazonia acidium. It is an inhibitor of the microtubule, so it is used as a anti-drug, anti-cancer drug. And some alkaloids and non-nitrogenous compounds are present in the Ascidians. For example, the nearly 300 alkaloid compounds have been isolated from the marine Ascidians, and more than 20% of the natural products derived from the Ascidians are nothing but the non-nitrogenous compounds. And there are some commercially available drugs, that is antimicrobial drugs, isolated from the Ascidians: enterocin, polycarpamine B, dihydrotryptophan, octopeptide, styrene D. Similarly, anti-neoplastic and anti-tumor compounds from the various species of Ascidians. You see the compound name, its composition, the species, Ascidian species, and its activity. Mostly they are the anti-tumor activity, the compounds from the isolated from the Ascidians. Apart from these antimicrobial, antioxidant, and anti-cancer drugs, there are some compounds extracted from the Ascidians which shows the plant growth regulatory activity. And some compounds isolated from the Ascidians are used to control the insect as a pesticide. Okay, see the Malaysian tulicate, Clavellina plicata, Eudistum obscurum, Diadenum mole, Atriolum robustum, Palusia species, Diadenum species, and Applidium species. So these are the species which produce the so many compounds which are used as a insecticides. Then anti-fouling compounds. Among the 52 numbers of genera of the Bioprosepcting of Ascidia, about 20 numbers showed the potential antifoulants, which is followed by the Stolobranchia and Filifobranchia. Then antioxidant activity from various Applidium species. And some Ascidians are referred as a candidate species for the novel genes. So searching for new compound, in addition to searching for new compounds or biological active compounds, some of the novel genes have also been produced from the Ascidians. The streamlined genome of Cyanoa intestinalis is a very common sea squid which is closely related to the vertebrates on the evolutionary tree. Nowadays, most of the regenerative studies are carried out using the Cyanoa intestinalis next to the planaria and zebrafish. Usually the planaria larva and the zebrafish are used as a model candidate species to study the regeneration process. Next to these animals, the Ascidian Cyanoa intestinalis has been used for the studying the regeneration property and regeneration process. See, this is Cyanoa intestinalis. It is a very interesting Ascidian, simple Ascidian, where the regeneration capacity is comparatively very high. And some other model species which are studied for their embryogenesis, aging, and regeneration properties, apart from this Cyanoa intestinalis: Cyanoa robusta, Cyanoa savigny, Palusia mammaliata, Halosynthia roretsi, Malgula aculata. And one more colonial Ascidian, Botylus closeri, is well known for the studies on whole body regeneration and stem cell research, the Botylus closeri. And this is one of the important database, that is Ascidian network for insight to expression and embryological data. You can visit this database. It is a major database system in the tunicate community, hosting and giving access to the genome or genetics and anatomical data on the Ascidian development. So what are the genes? What are the genetic materials? how they express, and all the informations you will get from this database. This is the first published in the 2010, and the database hosts and gives the easy access to the genomic information from the tunicata. Okay, so far we have seen the bioactive compounds or pharmacologically important compounds isolated from the Ascidians. Apart from these biological active compounds or drugs from the Ascidians, there are some Ascidians which are edible species. Okay, we will go there. We will see that. There are some edible Ascidians: Microcosmos, Sulcatus, Styla applicata, Polycarpa, Halosynthia. So there are so many edible Ascidians, and the edible part, that is a test covering or the mantle body. So in the Japan and in the Korea and China, the edible part, that is the soft bodied part of the Ascidian, are used as a delicious food, and in some countries the Ascidian materials are used for the preparation of pickles. See, this is the Halosynthia roretsi at the fish market in Tokyo. So in the fish market you can see the Ascidians, edible Ascidians, Halosynthia roretsi. See, this is the Pyura siliensis. It's a dried bodies of Pyura siliensis. It is available at a market in the Chile country. And some processed tunicates for local consumption and exports. See, the dried Halosynthia aureum, which is readily available for sale in Japan. You can see the dried material of the Ascidian for the sale. And here, this is the canned Ascidian, Pyura siliensis from the Chile country. You see the squids. It's a canned one. It is for edible purpose. And this is the imported frozen Korean Ascidian, that is a Halosynthia roretsi, at a Korean market in the USA. The Koreans are exporting the Ascidian food to the various countries, particularly to the USA. And here the frozen Styla applicata, another simple Ascidian from the Korea, which is regularly exported to the USA. And here it is a dense clump of harvested Pyura siliensis. You see, it's a Pyura siliensis. From this, many soft-bodied animals are collected. And see, the Pyura siliensis is celebrated on a postage stamp in the country Chile. You see, this is a postage stamp of Chile country, where you can see the pictures of the Ascidians. So that much importance they are giving, the Chile people giving to the Ascidians. You see, this is a Russian. They are harvesting the Halosynthia aureum through the ice in the Providencia region in the Russia. They are collecting the Ascidians from the ice regions. And there are different varieties of cooked Ascidians served at the shopping complexes in the Japan. You see the cooked and fried Ascidians. You see, this is the red sea squid that can only live in the cold sea, cold water area only it will be available. It contains a lot of nutrient-rich sea extract in the harsh and clean sea. You see, it is consumed as a raw in the various countries. And it can be eaten as raw material, slightly boiled or pickled. It is said to be the rare sea animal combining the four tastes, that is sweetness, saltiness, sourness, and acidity. So if you consume this species of Ascidian, you can feel the four different kinds in a single. And some seafood market in Japan, they are selling the sea pineapple, nothing but Ascidians. So here in Japan, the Ascidians are cultured in a farm. It is called Ascidian farm, similar to the aqua farm. In aqua farm, some fishes or prawns are cultured. Similarly, in Japan and in other countries, Ascidians are formed in the marine ecosystem itself. So this is the dried flesh of the Ascidians, and you can see the Ascidians used as a side dish, and this is Ascidian pizza. some other forms of the Ascidians, and this is the Ascidian pickle prepared by the Dr. Tamil Shelvi. Right. So this is bioprospecting of Ascidians for the new kind of food with the high protein and nutrient valued content. Okay. Then apart from this, high throughput screening of Ascidian resources. So Ascidians can also be used for the high throughput purposes. So for example, the diversity of Ascidian species that is equal to chemical diversity, because there are a diverse form of Ascidians, and each Ascidian producing the different kind of chemicals or drugs. So all these drugs may be called as a chemical diversity. A major obstacle is that the many resources are wasted in pre-purifying and recharacterizing non-novel compound only to discover. So there are so many resources we are wasting to isolate a novel compound. So isolation of this novel compound with the biological potential activity is not a simple one. There are so many processes. So it is a laborious, it is a long duration process. So many samples may be wasted. Okay. For example, time consuming, it is a laborious, expensive. So we are spending a lot of time and lot of money in investigation of Ascidians in the process like sampling, cultivation, extraction, fractionation, purification, and structural elucidations. So in all these processes, we are spending a lot of time and money. But there is a process called de-replication. This is a new strategy which involves the various separation and analytical techniques, that is high resolution mass spectrometry. By adopting this high throughput technique, one can easily isolate and develop the drugs from the Ascidian extracts. So this is a high throughput technology for the isolation of the different novel compounds. Now we come to the conclusion part. From this brief overview, in which I have summarized the current state of the art and the prospect of for the discovery of new bioresources from marine Ascidians, it is evident that several success stories have already emerged, and that interest in the field is likely to grow away beyond what has been seen in the previous years. The bioprospecting, the immense Ascidian biodiversity could lead to a new ways to address number of today's global issues, including the COVID-19. So the Ascidians have long been considered, earlier, the Ascidians have been considered as an organism of nuisance value because they are the good biofoulers. So a large amount of Ascidians they attach on the hull of boats, hull of ship, and many horrible installations. So the fishermen and the port authorities, they feel the Ascidian as a nuisance one, because they have to clean the bottom of the boat, hull, and other horrible installations which will disturb the functioning of the port. So they feel the Ascidians as a nuisance value. But the promising works on the recent years on Ascidians has raised the status of these organisms to a level where they are being viewed with favor in biotechnologically relevant spheres. So many bioactive compounds with the anti-cancer drugs, antiviral drugs, antibiotics, anti-fouling drugs, even anti-COVID drugs have been isolated using the Ascidians. So this recent investigations towards the bioprospecting of Ascidian has raised the status of the nuisance value into a biotechnologically significant species. So with this, I finish my presentation. This is the acknowledgement There are some photos, pictures, and informations I collected from the net sources, and all these sources have been acknowledged here. Thank you, Nodal.
Thank you, sir. Thank you very much, sir, for your efficient speech, sir.
Okay. If anything you want to clarify, if you want to ask anything, you can ask, and I'll be more happy to explain.
Students have any doubts about Ascidian? He is the correct person to clarify your doubts about Ascidian. Girls, are you interested in asking questions?
Sir, how can we clarify and identify the difference between Ascidian and sponge, sir?
Yeah, it is a good question. Actually, please mute your mic. Yeah. So since asked the questions, how can we identify the sponges and the Ascidians in the field? It is very difficult to identify, discriminate the sponges and Ascidians in the field itself. But there is some methodology by which we can differentiate. So if you touch the sponges, you will feel some rough surfaces, and there will not be any pumping of water. So we can identify it as a sponges. Whereas in these Ascidians, if you touch the Ascidians, you can feel some smooth surface, and if you touch or if you disturb the Ascidian, it will squid some amount of water. So this is the basic field technique to differentiate the Ascidians from the sponges. In some cases, Ascidians will be in the sheet form. Near to this Ascidian, the sponge also will be in the sheet form. So it is very difficult to identify or discriminate the sponges and Ascidians. But this is the way by which you can differentiate. If you touch the sponges, it will be very rough in nature. Okay, it will not pump out any water. It will not squeeze any kind of water. Whereas if you touch the Ascidian, it will be smooth and it will pump some amount of water. So this is the identification technique in the field, so that we can differentiate.
Thank you, sir.
Thank you.
Sir, some chemical compounds are present in the Ascidian, you told, no, sir, for anti-drug and anti-cancer drugs, and these will be present in the skin or in the fluid present in the Ascidian, sir.
Yeah, okay. So some compounds like anti-fouling compound will be present in the test, that is the outer surface area of the animal. Whereas some antimicrobial drugs or antiviral drugs or any other compounds which will be present in the soft-bodied animals. Okay, so actually those who want to isolate the bioactive compounds, they will collect the samples and the whole animal will be used for the extraction. But in the case of solitary Ascidian, that is a simple Ascidians, we can remove the test and the mantle body separately. Whereas in the colonial Ascidians, you cannot remove, you cannot separate the test and the zooids. It is very difficult in the colonial cases. So in the colonial cases, the whole animal will be used for the extraction of the compounds. Whereas in the simple Ascidians, you can use as a whole animal, or you can separate the test and the mantle body for the extraction, so that you will get the different kinds of compounds. Some compounds will be present only in the test, particularly the anti-fouling compounds. Anti-fouling compounds will be in the test part, whereas in all other compounds will be in the mantle body part. Okay, so based on the target of your research, you have to plan accordingly. You have to choose the materials accordingly.
Okay, sir.
Yeah.
Any more questions, ma'am?
Any other questions?
Okay, sir. Anti-fouling, what are anti-fouling compounds, sir?
Oh, there are nitrogenous compounds. There are so many nitrogenous compounds which shows the anti-fouling compounds. but the actual compound name I have not referred, but 22%, 20 more than 20% of the bioactive compounds isolated from the Ascidians are the antifouling compounds. They are belonging to the nitrogenous compounds.
Okay, sir.
Yeah.
Sir, while extracting the needed material from the Ascidian, or it will affect it, or we can safely remove it.
Pardon, I can't get your question. Please repeat.
While extracting the needed material from the Ascidian, sir, or it will make it, how can I?
Safety. You are saying about safety of. Is it
It
so?
will lead to
Fatima?
death or some other impacts on the Ascidian.
Ascidian may be affected or not. She's telling.
Ascidians will not affect. Will not be affected. We are collecting the Ascidians for our own purpose.
Because we are collecting more and more numbers.
Yeah. Okay. I got it. So when we collect the large number of Ascidians, the species may become extinct or the population will be depleted. Right. This is the question. Is you asked now?
Fatima, is this your question? Fatima, friend, is this your question?
No, ma'am.
During extraction அது எதுவும் நடக்குமான்னு கேக்குறா சார். எக்ஸ்ட்ராக்ட் பண்ணும்போது எதுவும் கையில எதுவும் பற்றுமான்னு கேக்குறாளா என்னன்னு தெரியல.
No, nothing will happen. But even then, we should be very cautious and we should use some gloves for the collecting because we don't know which Ascidians will secrete some chemical, dangerous compounds that we do not know. So it is better to use the gloves for during the collection. But so far we have collected large number of species without using the gloves. Nothing happened to us. So if you are worrying about in that angle, you use the gloves. Nothing to worry.
Any other questions,
And,
Pa?
Girls, any other questions? Okay. Shall we proceed? Anybody? Okay. Radha, you proceed. Radha.
Okay. Now let me propose a vote of thanks on behalf of our zoology department. First and foremost, I express my heartfelt thanks to our managing board members, our principal, Dr. Meena Rani, who gave us this precious opportunity. I extend a very hearty vote of thanks to our resource person and my PhD guide for his very informative lecture. Sir, today you have explained a very new concept, but in a very simple manner and in a very clear way. You have explained concerning the level of our UG students. You gave a very nice and interesting presentation, including the photos. The data collected were very informative and very updated. Definitely our students are very lucky to hear your speech, sir. We are also very much excited to hear your speech, sir. Thank you very much, sir.
Thank you, ma'am. Thank you, all of you.
Thank you, sir.
Thank you, sir. We extend all the hearty thanks to our HOD and all the other staff members in our department who all supported us, and also we thank the DPT scheme by which we have gained the financial assistance for this very wonderful program. Thank you, sir. Thank you.
Thank you, one and all. Thank you all.
Thank you, sir.
Yeah. Okay.
Thank you, sir. Thank you very much, sir. Okay, sir. Okay, sir. Thank you, sir.
Students,